All right, thank you very much for the opportunity to be here. My name is Jeannette Osterloh. As was mentioned, I'm a postdoc in Steve Finkbeiner's lab at the Gladstone Institutes. We're a nonprofit academic institution associated with UCSF. And today I'm going to be telling you about Daedalus Bio, which is a entity that we've spun out of Gladstone focused around the development of therapeutics for neurodegenerative diseases. All right, so we're interested in looking at targets for brain diseases. These represent a very large and growing financial and personal burden. In 2012, it was estimated that about 20% of the U.S. population was affected at some point in their lifetime by a brain disease. About half of these were an adult-onset neurodegenerative disease. So these are diseases like Alzheimer's, Parkinson's, ALS, or Huntington's disease. These diseases cost in direct medical care about $500 billion a year in the US alone. And when you take into account indirect costs, such as lost time at work, these costs nearly doubles. So despite this huge need and burden, we don't have really any disease-modifying therapeutics. And this isn't for lack of trying. We make a lot of drugs that pass the preclinical tests, but once they ultimately go on to human trials, they fail. And when you look broadly at failures for class 2 drugs, most of these drugs fail because of lack of efficacy. What this indicates to us is that there is a fundamental flaw in the preclinical pipeline, specifically when treating neurodegenerative diseases. So this is a little surprising given the complex and rate of nature of the preclinical pipeline, but we at Daedalus hypothesize, if you will, that the major failure for this pipeline has to do with its dependency on rodent models and cell lines. So at Daedalus, we are changing this preclinical pipeline and putting the human at the center and first and foremost for all of the preclinical developments. So we do this using 3 key pieces of technology. The first is that we harness iPS cells specifically from patients with the neurodegenerative disease that we're interested in studying. And then we use a variety of differentiation protocols to generate the brain cell type that's affected in the disease. This allows us to look for disease-relevant phenotypes, which so far in all of the familial forms of neurodegenerative diseases that we've looked at, we've actually been able to identify disease-relevant phenotypes from iPS-derived neurons. This also allows us to make isogenic controls for these and provides a cellular platform for unbiased genetic screens and small molecule screens, looking for modifiers of these disease phenotypes. The other key piece of this iPS technology is that it allows us to model sporadic disease, which accounts for the vast majority of neurodegenerative diseases. And this is something that we can't do with the current rodent models. The second key piece of technology harnesses whole genome sequencing and whole genome analysis. So we identify families that harbor our disease mutations and also have family members where the disease onset is very different. So there's at least a 7-year difference for onset. And so we comb through using whole genome analysis to look for specific modifiers. So for the example here, this is one of our Huntington's families. You'll see our initial patient had an age of onset at 56 years old. They have a sibling that also harbors the Huntington's mutation, but at age 70, they're still not symptomatic. So we would hypothesize that this patient harbors some sort of protective variant within their DNA, and we're looking to uncover that. Conversely, this patient's child began showing symptoms of Huntington's disease at 30 years of age. So we would predict that this patient then has either lost some protective variant or has picked up a deleterious one. And so we do whole genome analysis to try to tease these apart. So we use these 2 technologies in combination to develop a top-down and a bottom-up approach for modeling the disease and looking for novel targets. And this allows us, again, to do this in a fully humanized system, so we don't use any rodent cells at all during this process. Now, what makes this technology really feasible is the third key piece of technology, and this was developed by one of our founders, Steve Finkbeiner, and this is called robotic microscopy. So you'll see a video here of our 4th generation robotic microscope. This has a robotic arm that pulls plates out of the incubator, delivers them to our microscope stage, and this stage is fully automated, as is the imaging. So once the imaging has finished, the arm will put the plate back in the incubator, and then we can return and image the same set of cells over and over again. So typically we image these cells live once a day for about a week. And what we end up getting are movies, which you'll see here. So we can actually watch neurons develop and grow out over time and monitor how long they survive during the course of an experiment. So we take all of these images and run them through custom automated analysis platforms, which identify cell bodies and assign them a unique barcode number. It then goes through and tracks these cell bodies during each different time point, and we can extract a variety of features from the fluorescent measurements. So our most basic measurement is we look at morphology and viability, but we can really measure anything that you can sense with a fluorescent reporter. So our favorite is to also look at levels and aggregation of specific disease proteins, but really the options there are only limited by your reporter. All right, so this is particularly suited for working with iPS because it allows us to track many, many individual neurons over time. And so we can get very robust data sets and very robust phenotypes using a relatively low amount of sample. This is particularly important given the heterogeneity and the cost of generating iPS-derived models. So I don't have time today to go into all of the things that we can do with robotic microscopy, but I thought I would show you an example of where we've been able to use iPS-derived neurons and whole genome analysis to identify novel modifiers of the disease. So when we look at morphology and viability, many of you are used to looking at Kaplan-Meier plots. We use a derivative of that where we look at Cox proportional hazards modeling. So it's essentially a Kaplan-Meier plot, but turned on its side. And so when we look at survival, we focus first on the red line. This is our wild type control line. And so what you see is the line is relatively shallow, indicating that we don't have a lot of cell death over time, or the cumulative hazard of death is relatively low. If you compare that first to the line in blue, this is our iPS-derived neurons from a patient with an expansion that has Huntington's disease, and you can see that the cumulative hazard is quite a bit higher, indicating that the risk of death is much higher in these lines. Using whole genome analysis from one of our Huntington's families, we were able to identify a novel variant, which for obvious reasons I can't disclose here. But what I can tell you is that when we knocked this down using siRNA, that's the line in yellow, we were able to rescue the survival defect and restore it to wild type levels. So this is really a validation of our platform, showing that we can use whole genome analysis to actually identify potential modifiers of the disease, and then follow up and validate them in the iPS model. Right. The other thing that's very unique about this platform is that we can do deep phenotyping with the iPS lines, and this is allowing us to uncover molecular differences between patients who have essentially the same clinical disease. And we think this is going to be very important for moving these targets and these treatments forward, because it allows us to actually stratify the patient population and identify which patients are most likely to respond to a given treatment. So then we only enroll these particular patients in the trial, and this increases the chance of success for the trial while lowering the number of enrollees, which reduces the cost quite a bit. All right, so to summarize, Daedalus Bio is a new entity out of the Gladstone Institutes where we're developing novel therapeutics for neurological diseases. The unique thing with Daedalus is that we're using a fully human platform, taking advantage of iPS technology, whole genome analysis on familial neurodegenerative disease cases, and robotic microscopy to uncover disease-relevant phenotypes. And finally, we can use these disease-relevant phenotypes to actually stratify patients so that we can increase our power once we go to clinical trial. So we're always looking for new collaborations and new things to test. We have quite a few already within the lab. These are a list of some of them here. But again, we're always open and willing to take on more collaborations because we're really excited about this platform and the potential for it. So with that, I'll say thank you, and I'll be available for questions for the rest of the day.